Alrighty guys, welcome to my video about mitosis. We're going to be learning general information required for the 2023 cohort for year 12 term 1 biology. Now what we're going to be looking at is why mitosis is important. First of all, mitosis is important as it allows the growth of multicellular organisms. If we didn't have mitosis, we wouldn't be able to get taller, get stronger, or gain bigger muscles. It does this by repairing damaged tissue, replacing worn-out cells, and allows asexual reproduction, which means reproduction with itself, with growth of plants or cutting and cloning animals. Those are examples of asexual reproduction. And it also has genetic stability, which is the precise and equal distribution of chromosomes to each daughter cells. This pretty much means that it produces identical cells that have the same chromosome number, genetic information as each other, and the original parent cell, because they're all identical. Now this is pretty much what mitosis is. We'll be looking through the stages of mitosis. Obviously, mitosis is interphase is what's going on 90% of the time. The DNA and such. Now, how does this work? In interphase, DNA replicates by separating into chromosomes. And because I'm very nice and very smart guy, I have everything open. Now, DNA replicates by separating into chromosomes, and then in prophase, the nuclear membrane breaks down. The chromosomes prepare to divide. They get thicker, shorter, and the spindle fibers form. This can be seen in such a diagram that looks like this. I'll put it right there. As we can see, they have separated into chromosomes. The DNA obviously has separated to chromosomes from a bunch of dots into these, and then it's preparing to divide. The nuclear membrane, that blue line around it, has started to break down, and spindle fibers are forming, which you can put as two dots and a couple lines branching off. Now, we have metaphase next. In metaphase, the chromosomes line up in the equator of the cell. Metaphase, middle, they line up in the middle of the cell. Spindle fibers attached to the centromeres. Centromeres are dots in between the X, pretty much where the X is like intercept, and that's what the centromere is, and that's what happens pretty much. They line up in the middle of the cell like so. We have a picture right there-ish. As you can see, lined up in the middle of the cell, spindle fibers are attached to the middle of them, which is where the centromere is. You'll be able to draw that by putting a dot right in the middle. Now make sure you label that as the centromere, which is obviously something that you have to label. In anaphase, think of anaphase away. They're pulled in opposite directions. So in metaphase, we can see that they are like so, and in anaphase, we'll be able to see them getting pulled in opposite directions because they've separated from chromosomes into chromatids, and now that they've been pulled in opposite directions, that's what they will look like. The spindle fibers are obviously still attached. Now in telophase, think about it. Telephone, you're putting it down, calls over, the nuclear membrane and nucleolus reappear. The mitosis is complete. But we have two nuclei and identical chromosomes, but the cytoplasm is still together. So we're going to be drawing that as something like this. That's what it'll be looking like. Mitosis is complete, the cytoplasm is still together. Those lines in the middle are spindle fibers that have broken down. Now in cytokinesis, we're going to end up with two of these, and that's pretty much what it is. The cytoplasm would divide, and we'd have two perfect circles with the daughter nuclei with the chromosomes inside it. And that's pretty much exactly what mitosis is. C. Remember that. That's also necessary in meiosis, which allows us to have less memorizing, but it's also a bit more confusing. So thanks for watching guys, and I'll see you in the next video.